Hello everyone, we are Group 18. Today, I would like to talk about minor histocompatibility antigens, or MHAGs. In transplantation medicine, we often hear about HLA or MHC matching. These are the major antigens. But today, I want to focus on the minor ones. This is MHAGs. Even when the major antigens perfectly matched, these minor antigens can still decide the success or failure of a transplant. First, let's define what minor histocompatibility antigens are by comparing them to MHC. MHC molecules are structures on the cell surface that hold and display small protein fragments called peptides to the immune system. If the MHC molecule structure itself differs between a donor and a recipient, the immune system attacks immediately and strongly. This is a major mismatch. However, even if the MHC molecules are identical, the peptides they display can be different. These peptides come from internal cellular proteins. Due to slight genetic differences between individuals, the amino acid sequence of these peptides can vary. These differing peptides are the minor histocompatibility antigens. They are called minor because the rejection happens more slowly than with MHC mismatches, but they can still cause severe immune reactions. Look at this animation. When MHC molecules and T cells receptors aren't matched, it will trigger rejection. This is a major factor in transplantation rejection. Even though MHC fits T cell receptor, if peptides which MHC presents are different from host peptides, it can lead to rejection. These peptides are MHAGs. Let's take our first example, HY antigens. Here we think about male to female organ transplant. Genes on the Y chromosome are transcribed and translated into proteins inside the donor's cells. These proteins are then naturally broken down into small peptides, the HY antigens, which are displayed on the cell surface by MHC molecules. To the donor, these peptides look completely normal, but to the female recipient's immune system, the HY peptide is entirely new. Even though the MHY molecules match, her T cells recognize the MHY peptides as non-self, and this mismatch leads to rejection. In male to female transplantation, donors' HY antigens can be target for recipient's T cells and lead to rejection. Now, let's look at a second example, graft versus host disease, GVHD. This occurs in the reverse scenario, such as a female donor to a male patient in stem cell transplantation. Here, we are transplanting the female donor's stem cells. They become two immune cells like T cells. The donor's T cells have never seen the HY antigen before. When they enter the male patient's body, they find the HY antigen everywhere, on the patient's skin, gut, and liver. The donor cells start attacking the patient's entire body. This attack is called GVHD, and it is a severe complication where the graft attacks the host. In summary, minor histocompatibility antigens are small peptides. They are displayed by MHC molecules. Even though MHC is matched, transplants can cause rejection and GVHD because of MHAGs. Thank you for listening.